So today we will be talking about the microscopic anatomy of the respiratory tract, both the conducting zone as well as the respiratory zone. We will talk about the functional histology of the entire system, which is really very important to be understood, because that way we will be able to understand how each element of the entire system is working. Because at cellular level, the functions have been performed. So it's really very important to understand the functional histology of the system. So first, let me talk about the general histological pattern of the airways. Here, have a look at this drawing. It's showing us the arrangement of layers, how the tissue inside the air tube is being arranged. So this is the inner lining of the respiratory tract, particularly the conducting zone. I'm talking about the lower respiratory tract, which starts from trachea downward till the terminal bronchiole level, and then we will talk about the respiratory zone, which will start from the respiratory bronchiole. So here, have a look at it. So if this is the lumen of the tube, the epithelium is the innermost layer which is coming in contact with the air. So we know that the epithelium of the respiratory tract in general is a specialized type or modified type of epithelium known as Pseudo stratifiedciliated columnar epithelium. The name is indicating it must be having some hair-like projections. Now look at the cells. Most of the 30 to 35% of the cells which are lining this epithelium are simple, they are long columnar type, like column-like cells with the cilia pointing at their apical end or luminal end. So every cell has a basal surface or the basal part and an apical part. The apical part is facing the lumen, and the basal part is resting on this white line, which is the basement membrane. This line is representing the basement membrane of the epithelium. Then there are 4 different types of cells present in a respiratory epithelium in general. Tall columnar ciliated type of cells, the goblet cells with no cilia. Why do we call them goblet cells? Because they are of the shape of wine goblets with the long stem and an expanded top part. So they look like goblets. This is how they look like. The 30% of the population of these cells is composed of the goblet cells, and the goblet cells at their luminal side, they are like, the cells are resting on the basement membrane, the goblet cells, and they're reaching up to the lumen, surface of the lumen, but the entire cell has been filled up by secretory granules. Those are the granules for mucus. So we need a lot of mucus in the respiratory tract, upper airway and the lower airway. So the goblet cells are the second type, and they do not have cilia on their luminal surface. Then the third type of cells, they are very dwarf cells. These are known as the basal cells, you can see. The basal cells are resting on the basement membrane, but they are never reaching the top. They are never reaching the lumen. These basal cells are said to be the stem cells. What they are doing, they are constantly multiplying and giving rise to new generations of columnar cells and the goblet cells. So they are the factory. Then there are cells which are dispersed within this line of cells. The type of cells are DNES, diffuse neuroendocrine system cells. Those cells will be responsible for the production and release of hormones. They are present in most of the epithelial linings of the respiratory tract, the gastrointestinal tract, and they are present in majority of the places. The diffuse neuroendocrine system cells. Then the epithelium, under the epithelium, you know, the epithelial cells are resting on the basement membrane. The epithelium itself is an avascular tissue. It doesn't have a blood supply. How these cells, they get their nourishment? through the basement membrane, which acts like a semi-permeable membrane. And what is lying underneath the basement membrane is a layer which is known as lamina propria. So together the epithelium and the lamina propria will make the mucosa, the inner lining of any tube, and in this case the respiratory tube. So the lamina propria in general in the upper airways is being thickly populated by the seromucous type of glands. So I just have mentioned that we need a lot of mucus. So not just the goblet cells, which are the factories, mini factories of the mucus production, but there are seromucous glands present within the lamina propria. Here you can see a population of blue colored dots. These are the white blood cells. They are the first line of defense for us. So the neutrophils are there. Then we have different types of lymphocytes. The T lymphocytes will also be forming lymphoid nodules that would be present in the lamina propria. Then you can see these capillaries, right? They are bringing the oxygen and nutrients. So through the lamina propria, the basement membrane will allow the nutrients to nourish these cells. Mind you, this is a single layer of cells, but it appears like under a microscope, it appears to be like multiple layered, because the levels of the nuclei are not at the same plane. Some nuclei, because the nucleus is the king of the cell, it requires a lot of space for its survival. And we know that there is overcrowding of cells. The columnar cells are, pushing each other. So the nucleus is always located in the expanded region of the cell. So we can say that the nuclei of the pseudo-stratified columnar epithelium is showing at different levels. That's why it's giving a pseudo appearance of a stratification. Alright, enough of the epithelium. Now, below the mucosa, which is composed of epithelium and lamina propria, what do we have? The submucosa. By the way, the lamina propria ends at a point where it is having a thick band of elastic tissue. Okay? An elastic lamina is separating the lamina propria from the submucosa, which is lying underneath it. Then we have the submucosa, which is in general composed of the fibroelastic tissue. The fibers are arranged in a haphazard fashion, like in a net, as a network in a reticular formation. And the submucosa is also having a decent population of the seromucous glands, which are releasing their ducts are opening into the lamina propria, and through the lamina propria they are reaching the surface, or the luminal surface of the tube. Then we have these clusters of lymphocytes. These are known as the lymphoid nodules, which will be present in the submucosa. Then we have the autonomic nerve fibers, which are here in the submucosa. And then these arterioles and venules are accessing the submucosa, and then the capillary network you will be finding in the lamina propria, and so on. Then if we come out, like this was the innermost layer, the mucosa, then in between is the submucosa, and the outermost layer is known as adventitia. Adventitia in general everywhere is composed of thick collagenous fibers, the connective tissue fibers. Here also it's having a huge collection of connective tissue fibers, and the blood vessels are approaching the inside of the lumen through the adventitia. This is a general plan, the microscopic plan of the respiratory lining. Now we have to understand this concept that as the airways are getting smaller and smaller in size, in caliber or diameter, they will be losing this arrangement, because they will be changing, transforming according to the demand of that tube. This was the general pattern. Now we'll move on to the other pictures, and I'll explain one by one. Alright, so now I want you to understand something. First of all, there's a table. So the trachea is the windpipe. It's the first part of the lower respiratory tract. Then comes the primary bronchi, right and left. These two structures or the tubes, they are considered to be extrapulmonary airways. They are outside, absolutely outside the lungs. The secondary bronchi, you must have remembered that on the right side, in the right lung, one of the secondary bronchi has been given off by the stem bronchus way before it enters the tissue of the lung. So that is an exception, but in general, we can say that from secondary bronchi, tertiary bronchi, till the level of terminal bronchioles, all of them are considered to be airways. They are the part of the conducting zone. So till the level of terminal bronchioles, the end of conducting zone, the tubes are considered to be intrapulmonary airways. Secondary bronchi till the terminal bronchioles, everything is intrapulmonary, lying within the lung tissue. And we know that the lung tissue is a very soft, spongy, it has a spongy texture. A healthy lung I'm talking about. So we can imagine and we can understand how things will be changing in these layers, the basic layers, the histological layers. We need to get rid of cartilage material, which is hard for the lung tissue. So I have given you a clue. Now we'll move on. We'll move on to this drawing. This is specifically is the drawing of the trachea. And we know that the trachea is a cartilaginous tube. It has the band of smooth muscle lying at the back of it to complete that C-shaped ring. But majority of the trachea has been formed by the C-shaped cartilages or the U-shaped cartilages. The cartilages are keeping this part of the airway open for the air all the time. So we have our typical pseudo stratified ciliated columnar type of epithelium, and I have forgotten to do something. This is the layer of mucus. This is the layer of mucus on top of the cilia. And I did mention in my previous lecture that the mucus or the cilia in the lower respiratory tract, they are beating, there is only one direction they are beating. That direction is upward towards the nasopharynx, so that the mucus, this thick film of mucus, should be pushed up. That is loaded with the debris and other foreign particles, and that will be moving like the lower airway, the lowermost region or the intrapulmonary airways. They are very sensitive to the particulate matter, the dust and other foreign bodies and organisms. They are very, very sensitive. The epithelium down there in the lungs is very sensitive. So we cannot afford to have those loaded, that loaded mucus to go down. So we need to get rid of it. So the ciliary movement in the trachea and the bronchi would be upward. All right. So pseudostratified ciliated columnar epithelium with a lot of goblet cells constantly producing the mucus. Then we have the lamina propria. The typical lamina propria would be evident. Then we have the submucosa. The typical submucosa is there. Then we have in the adventitia, we have something which is not, I didn't talk about it. This is a section through hyaline cartilage. So the adventitia is housing those cartilages of the trachea. So what happens sometimes, people who are chronic smokers, they just lose the mobility of the cilia. Okay, their cilia get immotile, right, and sometimes they degenerate. So that will, allow the mucus to go down towards the lower airways, and it heads towards the lungs. Why? Because the cilia are not performing their function and the mucus is constantly being produced. In fact, in the smokers, in chronic smokers, there is excessive production of mucus. Why? To get rid of the smoke, which is known as soot. Those carbon particles, they just want to, the system just wants to get rid of that solid particle. So it produces more and more mucus. And at the same time, the excessive mucus doesn't have a way to go up towards the nasal cavity or towards the nasopharynx because the cilia have stopped functioning due to chronic smoking. And sometimes what happens, this layer, the epithelial lining of the mucosa, it just due to constant irritation for a very long time, it shows some metaplasia. There is change in the type of epithelium. There would be overpopulation of goblet cells, as I just have described you, that they require to produce more mucus, so there will be more goblet cells, and sometimes the columnar cells will be, eventually be replacing themselves with the stratified type of epithelial cells, because there is too much irritation in the upper airway, the trachea. Okay, so we know that at the level of trachea, in the adventitia, we have the hyaline cartilage, a continuous bar of cartilage is there. As far as the primary bronchi are concerned, they have the same pattern. They just imitate the trachea because they are just the extensions of trachea, and they are still lying outside the lungs. Then at the level of secondary bronchus and the tertiary bronchi, this continuous band of cartilage is not visible. The cartilage would be present in chunks, a little here, a little there, not a continuous band. Then the tertiary bronchi will end up into bronchioles. The bronchioles are the tubes of not more than 1 millimeter diameter. Any tube which is having a 1 millimeter diameter is known as bronchiole. What is happening at the level of bronchioles? You can see over here, there is a change in the epithelium. So as we are going down, the pseudo stratification is disappearing. At the level of tertiary bronchi, or the beginning of the larger bronchioles, the epithelium will show like a population of single low columnar epithelium with less number of cilia. As we come down to the very small bronchioles, the epithelium will be changing. The cells, the heights of the cells will be further reducing from the columnar, low columnar to cuboidal. And there will be again a single layer of cells. Cilia will be just occasionally present here and there. The population of goblet cells will be completely gone. There won't be any goblet cells at the level of bronchioles. Then at the same time, the lamina propria will reduce in thickness. There will be hardly any glands. There are no glands because we do not require any secretions in the lowermost airways, the intrapulmonary airways. We do not need any secretions. So we do not need glands. We do not need mucus. So there are no goblet cells. Instead of goblet cells, you can see something which is populating this epithelium at the bronchiolar level. These are known as the Clara cells. The Clara cells, sometimes they are also known as the club cells because they are of the shape of club. So the Clara cells, as you can see, they have fine granules in them. Some books, they say that the Clara cells are responsible for the production of a surfactant-like material to keep the bronchioles open, to keep them open for the passage of air, because we do not have any cartilage material here, and the number of smooth muscle fibers will also be getting reduced. So we need a lot of, surfactant-like material. So the Clara cells are also, some studies have suggested that the Clara cells, they have certain granules which are helping in detoxifying. So they act like defense cells also. Then at the junction between the lamina propria and the submucosa, we have thin bands of smooth muscles fibers. Here I have drawn. So this is a terminal bronchiole. So you can see there is the bands of the smooth muscles, they are crisscrossing each other. They are running in opposite direction. They are doing what? They are for the expiration and inspiration, we need the movement of the tube. Okay, so they are responsible for bronchoconstriction, then bronchodilation. So these are the muscles I was talking about when I was discussing the innervation pattern of the lungs, lungs and airways, that the smooth muscles of the bronchi and bronchioles are under the control of sympathetic as well as parasympathetic stimulation. So we know that the lamina propria and the submucosa, they have an intervening layer of bands of the smooth muscle which are running in a crisscross fashion. There usually are two bands. So what happens that sometimes people who suffer from asthma, normally there is these muscles in the bronchioles, they contract at the beginning of expiration. And then during inspiration, they get relaxed and they allow the air to gush in. People who are suffering from asthma, what is happening with them, that the bronchioles, the musculature of the bronchiole just stays contracted for a very long time during expiration, right? So the lumen of the bronchioles, the lumina are still closed, and the people are really suffocated. They feel suffocated because they cannot inspire air because of their prolonged contraction, bronchoconstriction. At the terminal bronchioles, we are done with the intrapulmonary airways. So you have seen how the things are getting different, starting from the epithelium. It started as a tall, columnar, pseudo-stratified type of epithelium with a lot of cilia and a thick mucus layer on top of the cilia. The lamina propria was loaded with and these changes are gradual, right? So the things are transforming, especially the epithelial lining is transforming itself according to the demand of that tube. So from the tall columnar, pseudo-stratification, it becomes the simple columnar type with no stratifications, and then ultimately low columnar will transform into cuboidal type of epithelium. The goblet cells have been replaced by the Clara cells. Cilia are just scanty, just here and there. No mucus production, especially beyond the bronchiolar level, like at the level of bronchioles, starting from the very large bronchioles till the smallest, that is terminal bronchioles, we do not see any form of glands or no production of mucus. That's not there. We need to keep the lower respiratory tract and especially the respiratory zone as dry as possible. Then we'll move on to the respiratory zone. That starts from the respiratory bronchiole. You can see that these bands of smooth muscle, they still are continuing themselves in the respiratory bronchiole. The respiratory bronchiole, the inner lining of the respiratory bronchiole will be pretty much similar as that of the terminal bronchiole. The number of Clara cells will be increased, and the simple cuboidal epithelium will maintain itself. Hardly any cilia will be there. The elastic fibers are there, and the smooth muscle bands are there. The only difference between the terminal bronchiole and the respiratory bronchiole would be the appearance or the bulging out of the walls of the respiratory bronchiole as the blind sacs. The appearance of alveolar sacs will indicate the change of terminal bronchiole into respiratory bronchiole. Then there are certain stages of like alveolar duct, then alveolar sacs, and ultimately the alveoli. Here are the alveoli. The bunch of grapes is known as alveoli. These are air-filled spaces. And as you can see over here, these are known as the interalveolar pores. And they are very important. Through these pores, the alveoli are having a maintained pressure in them. They are communicating with their neighbors. And these are known as the interalveolar septa. Interalveolar septae. Just within the interalveolar septal zone, the capillaries are very extensive, blood capillaries. So when we talk about the cell types lining the alveoli, I have discussed it when I was doing the embryology with you, that two types of cells are evident. The type 1 pneumocytes, which are the smooth, they are the simple squamous type of cells. The cuboidal cells, they have replaced themselves with the simple squamous type of cells with the cytoplasmic processes attenuated, very thin, to allow the exchange of gases. So type 1 pneumocytes, and then there are type 2 pneumocytes. You can see they are larger cuboidal cells, and they have granules and lamellar bodies in their cytoplasm, the bodies which are constantly responsible for the production of surfactant, because these are the surfactant cells, type 2 pneumocytes. They are also known as septal cells sometimes, because they are more in population at the point where one alveolus is meeting the other alveolus and the interalveolar septum is there. So these type 2 pneumocytes are more in number as compared to type 1 pneumocytes, and they are bigger, pretty much obvious in appearance, and their cytoplasm is having lamellar bodies, and they are responsible for the production and release of surfactant, which will be, covering the inside of each alveolus as a thin film of protein. What are these cells? There is a third type of cells present in the alveoli. These are also known as dust cells. You must have heard about them. Dust cells. So the dust cells are nothing but the alveolar macrophages. What they are doing, because we do not have any cilia, we do not have any Clara cells, we do not have any type of protection present inside the alveoli. But still we can get a lot of infected air. So the dust cells are the alveolar macrophages. They are present here and there, within, like inside the alveoli. And what they are doing, they are phagocytizing the foreign particles. Sometimes what happens, a person who is suffering from heart failure, right heart failure, or the left heart failure in the long run, what would be happening? There would be too much pressure in the pulmonary circulation, over the pulmonary limb of circulation. So that will cause the leaking of blood. And the old blood cells, like the red blood cells, they will ooze out of the capillaries, and then they will get deposited in the alveoli. These alveolar macrophages, this is their function, to eat up all the foreign particles, all the foreign elements. So they will be loaded with the red blood cell particles. So at that time, if you observe the lung tissue under microscope, the lung tissue of a person who is suffering from heart failure, you can call these the presence of these big macrophages, loaded macrophages, their cytoplasm is completely filled with particles, RBCs, broken RBCs, you call them heart failure cells. So they are nothing but the dust cells, the alveolar macrophages. So I just wanted you to appreciate how this transition has been taking place from up, like this, the things start from the nasal cavity. When I was discussing the nasal mucosa was having, the vestibule was having the stratified squamous type of epithelium, then the majority of the nasal mucosa was the respiratory type, just except for the top where the olfactory epithelium is lining the root of the nose. Then we have down in the nasopharynx, at the back of the posterior nares, we have nasopharynx, which is also lined by the respiratory mucosa, the pseudo stratifiedciliated columnar type of epithelium, with a similar lamina propria, a lot of lymphoid tissue. Then in the oropharynx and the laryngopharynx and the larynx, so oropharynx, laryngopharynx, they have a stratified squamous type of epithelium. Then in the larynx also, I just told you that above the level of vocal cords, the larynx is covered by the stratified squamous type of epithelium, because there is too much possibility of wear and tear, and too much friction is there. The vocal cords are also covered by the stratified squamous epithelium. But below the level of vocal cords onwards, we have pseudo stratifiedciliated columnar epithelium, that is the typical respiratory epithelium. At the level of bronchioles, this typical pseudo-stratified ciliated epithelium is gone by the time it reaches up to the level of bronchioles. It's been replaced, first it's been replaced by the short or the small columnar cells, single layer of columnar cells, very few cilia, no goblet cells and no glands. Then the low columnar epithelium is further replaced by the cuboidal epithelium, cuboidal cells. at the level of respiratory bronchiole, the terminal bronchiole, respiratory bronchiole, all of them have the cuboidal epithelium. By the time the alveolar ducts start, that cuboidal epithelium further gets smaller in height. So it is getting short cuboidal, no cilia, no glands, no goblet cells. Even once it reaches the level of alveoli, even the Clara cells are missing. No Clara cells, because the surfactant has been produced by the type 2 pneumocytes, so there is no need of Clara cells. The responsibility of defense has been taken care by the alveolar macrophages. So I hope that you have understood the basic functional histology of the respiratory system, and I hope that it will help you.